Next speaker, the 135 slot, which is Martha Grover, also from Georgia Tech, and she'll be talking to us about cycles and systems, ratcheting towards life.
Hi, everybody. Great to be here. I'm just going to assume you can all see me in my screen, and you'll let me know if there's any issue. Great to be here today, and great to talk about the work that I've had the privilege of being involved in over the last 10 years with my colleagues in the Center for Chemical Evolution. So similar to Facundo, a little over 10 years ago, Nick called me up and asked if I would be interested in research in the Center for Chemical Evolution, and I said yes, of course. I would be very interested in that. So I'd just like to start and mention one of the quote Ram referred to yesterday. I looked it up from Francois Jacob, 1977. Nature is a tinkerer, not inventor. I noted the engineer because I'm an engineer. I'm a systems engineer. But in any case, while true, as an engineer working in the origins of life, the perspective that I bring is that life is a complex system, and its origins probably were also pretty complicated, probably not a single reductionist sort of question. And so I've been able to bring some of my approaches and tools to this question over the last decade. One thing you've heard a lot about, and we'll hear more about, is about environmental cycles. And so it could be that if you have an environmental cycle, you start and you go around the cycle and you go right back to where you started. So that's possible. Not all cycles are productive, but sometimes through a cycle you can actually ratchet forward. You can take maybe one step back, but then 2 steps forward and actually move maybe in a new direction or a desired direction. So that's a thought that we can continue thinking about throughout the presentation. So the ultimate goal, I think in the CCE, but my goal in working in the center was to design and to demonstrate minimal systems that showed features of chemical evolution that could polymerize information, polymerize to store information, transfer that information, catalyze reactions, and ultimately undergo selection and evolution. So ideally, we would like to find a common environment in which all of these steps could occur. That's not necessary, but is ideal. And so far, I haven't really mentioned what kind of polymers we're talking about, but nucleic acid polymers, peptides, in particular, the areas that my work has focused on, but also these similar questions for lipids and polysaccharides. And there are some outstanding problems in chemical evolution that we have been considering within the center, and I think have gained some insights into. One you've heard about already in the symposium is the water problem. Biopolymers are formed through condensation reactions, but those are not favored in water. Another problem, longstanding problem, is the strand inhibition problem, and Jack Shostak talked about that yesterday. Heating can drive duplex separation, but cooling can lead to just going right back to where you started in that cycle. And finally, the single winner scenario. We know that life is complicated, it's made of diverse components, and we need diversity for higher level system function. But there are many models in the origins of life that show that the fastest replicator takes over the population. That doesn't lead to a productive outcome. It doesn't enable open-ended continued evolution, which requires diversity. So those are 3 outstanding problems. And we've been considering environmental cycles as one component in constructing these minimal systems that demonstrate chemical evolution. In particular, that we need to have different types of events that happen, sometimes during cold period, sometimes during hot period, wet and dry. And we would like these cycles to be prebiotically plausible. So driven ultimately through the influx of solar energy on a rotating planet, and that heating and cooling can then drive hydration and dehydration as well as other environmental cycles. Lunar cycles can also drive this sort of environmental cycle. But in particular, we've been considering cycles that have a particular directionality. So cold and wet occurring together and hot and dry occurring together, and then the heating really driving the hydration. And so that's actually going to be important going forward in what we're going to be talking about. And this idea of environmental cycles is not a new one within our center in the last 10 years, but has been discussed by Orgel and Wahab and many others. So early on in the center, almost 10 years ago, postdoc Sarah Walker was working with Nick and me on defining a mathematical model to take elementary physical and chemical events and show a phase space or parameter space in which we could achieve chemical evolution. And so in this particular model, we had a dehydrated phase, and then we had a hydrated phase, and we had reactions in the dehydrated phase driving both polymerization as well as replication. And then in the night phase, we had hydrolysis, that reversibility, back to monomers, as well as diffusion, limited diffusion of the monomer and the polymer. So this is a relatively simple model, but had chemical realism to it, in which the rate of polymer formation depended on the local resources. So that was an important feature we wanted to build into our model, kind of an ecological sort of effect. And we were able to show a couple of things with that model. One thing that we were able to show is that that possibly the first functional biopolymer could have been the monomer synthetase. It could have been a polymer that made more monomer, right? So generated more resources. So that was the hypothesis for this work. And what we showed is that if we just had these replicating molecules forming clusters, eating local monomers, breaking back down, we were kind of limited in our how many polymers we could have because of this limited resource. But if a functional sequence came along that could make more of one type of monomer, A, we could then really build up a larger population. But now we had too much A and we were limited in B. So then if at a later time and a later location we could make more B, now we could really bootstrap up the system to a much higher level where we could get full resource utilization. So this showed how we could have cooperativity emerge over time and space. We didn't have to suddenly form both functions at the same time to form this hypercycle that is thought to be important and minimal feature in the evolution of life, but it could happen stepwise. So through this study, we realized a number of things. One thing we realized is that there were a lot of sweet spots in the parameter space. We wanted some reversibility in our polymerization, but not too much so that we didn't forget everything every night when we had hydration. So we wanted to have some heredity. We also wanted limited diffusion so that we could have basically like niches where a functional sequence could benefit from its own resources, local resources, and its own function, but also we could share those resources. So we needed not too much diffusion, but not too little diffusion. We showed how these cooperative hypercycle networks could emerge stepwise. And then we also saw emergent behavior. Even though this was a very simple set of elementary reactions, we saw this emergent clustering that we didn't actually expect because we didn't build in any sort of association between the polymers, but because they were breaking down and then eating each other basically after they broke down, it led to this clustering phenomenon that we really didn't expect. So based on this study, we took away from it some features that we should put into our system design in the experiments that we were building. We wanted some backbone reversibility, so that would allow monomer recycling and more search through sequence space. We thought the environmental cycling would be important so we could drive condensation polymerization at some points in the process, but then have hydrolysis at other points in the process, as well as for strand separation and replication. We also wanted to have limited diffusion. We thought the diffusional effects would be very important in addition to the covalent bonding, that these sorts of mass transport effects would also be important to build into our system, and ultimately wanted to achieve selection based on function. So that led to this particular picture of a cycle in which we would start off with free monomers in a warm, wet solution, but it would dry and concentrate and form polymers through hydration, through dehydration reactions. But then it would cool down at night and rehydrate, the dew would form, and that would lead to certain sequences forming structures that would be protected then from hydrolysis, and so we could select for folded and functional polymers. So Facundo just talked about the esters, the hydroxy acids, And again, this was an idea that had been put forward by Rich and Orgal, but it hadn't been followed up so much. Many thought that the esters were not stable enough. But Nick really thought that we should have another look at the esters as the center was starting. And so we started looking at polyesters as a possible precursor to peptides and the amide bond. And as was previously mentioned, peptides will form this diketopiperazine that's very stable, cyclic dimer, and it's kind of a dead end. But with the esters, it'll open back up again. And in fact, you can have ring opening polymerization for forming polyesters, but we still would have the same side chains as we have for forming peptides. So that seemed promising. And so postdoc Irina Mamedunov, who's now a faculty member at LC, led a team of undergrads, and they performed these environmental cycles of malic acid and characterized the concentration of various species, monomer, dimer, and longer species over time at different temperatures. And we were able to build a polymerization model that just had a few rate constants for a chain length independent polymerization and a mass transfer constant, and to show how we could ratchet forward in the first few cycles, although then after that we were had this cyclic steady state. So then we didn't really progress. So first we had kind of a ratchet, and then we fell into this cyclic steady state. And so this was a paper that we published in Macromolecules in 2014. This is, I think, an example of sort of how fun the research has been in the CCE over the past decade. If you read this paper, you'll see this. We don't say anything about amino acids or ester-amide exchange. That was never part of the narrative. Here we were really looking at polyesters as being an earlier version of peptides. Then later, after life took over, then it could evolve somehow into peptides. But we didn't really address that in this paper. But as Facundo just pointed out in the previous talk, we were all having a discussion in the meeting, and Ram did suggest, let's add some amino acids and maybe we'll get ester-amide exchange, and we did. So Jay had just talked about the paper by Forsyth and Yu, in which we demonstrated that we could form these copolymers called Depsipeptides. And Sheng, who's a chemical engineering graduate student, developed a pretty detailed kinetic model of the copolymerization reaction, and he quantitated 9 different species, different oligomers, and fit a pretty complicated, computationally intensive model that had only really a small number of rate constants. So there were rate constants for 2 for polymerization, 1 for exchange, and then we had a mass transfer coefficient for lactic acid evaporation, and was able to show very good agreement and really very classical, even Arrhenius sort of dependence on these rate constants, which we didn't expect at all because these are drying reactions, and so they shouldn't really be elementary reactions. But in fact, they did appear to be modeled really using very basic chemical and physical events. And from that, he could construct this free energy diagram that again shows, if you wanted to form the peptide bond directly through drying, you'd have to go over this higher energy barrier. But by kind of going forward and backward and forward and backward again, we could actually move to a much more facile pathway for amide bond formation. And this work is continuing with the work of PhD student Calvin Smith, who we're collaborating with Charlie Leota on this work, and are really looking at the effect of pH on the ester-amide exchange reaction and the critical role of pKa, the fact that these reactions occur somewhere between the pKa of the glycolic acid and the glycine. That's really central to understanding the thermodynamic driving forces for this reaction. So some of the insights so far that we've gotten on the polyesters are that although hydroxy acids could have simply played the role of amino acids in prebiotic chemistry, it may be that they're the catalyst for the amide bond formation instead, or maybe both. Maybe the Depsipeptides were an important evolutionary intermediate, eventually leading to takeover by the amide bond. And then another realization that although the water problem has been long articulated in prebiotic chemistry, perhaps we use condensation polymers because they can be broken down in water. We don't want to just get into this dead end where we're just going around and around the cycle making diketopiperazine and then holding it there, that we really need to be able to move backward so that we can then move farther, ratchet farther forward. So that work on Depsipeptides is really about the ester bond as a backbone. That's important, but another long-standing problem in origins of life chemistry is the strand inhibition problem. So let's say you have RNA. So yesterday, Jack Shostak talked about how to polymerize nucleotides, but there's also the strand inhibition problem that Jack talked about, which is we can heat up a duplex, and then if it's in the presence of monomers or oligomers, we could form a copy, but in fact, when we cool down, that doesn't really happen. Instead, we just go back here. Okay? So that issue was articulated by Jack Shostak in a paper in 2012, and more recently work from his group and from Dieter Braun's group have looked at various strategies for overcoming the strand inhibition problem. But many of these approaches look at short sequences and unstructured sequences. And so we've been looking over the last 10 years at an approach that's pretty general, that also works for structured and long sequences, and that all of these approaches could potentially be used together as well. But the basic idea for this approach is that while we heat the duplex, we cool it in non-aqueous solvents or mixed aqueous and organic solvent, and that allows us a window of time in which we can coat the long strands with the short oligomers to make copies, and we've gone around the cycle now multiple times and shown that we can really ratchet that forward. So I'd like to, for the remainder of the talk, focus on the work of our viscosity team. Nick and I have been able to work with this talented team of researchers, and some of the seminal papers here are published. Chiamaka Obinyor is a current student who recently published a study on non-enzymatic DNA ligation, on the chemical ligation. I'm going to talk to you more today about using the alternative solvents, and then finally Bryce is taking us all forward and putting it all together. So again, we're looking at environmental cycles in which when we heat the system, the water would evaporate and we would have a low water activity, possibly high viscosity solvent. And the solvent that we initially chose to focus on was glycerol and choline chloride, or glycholine. One of the reasons we use this solvent is that it has been used previously. It's a deep eutectic solvent, and it's been shown that DNA retains its B form in glycholine, as well as other similar types of solvents like raylene from urea instead. But one of the nice features is that it's miscible with water, and so we don't really have to worry about different phases and phase separation, that the solvent is miscible with water, and DNA B form is retained. And one of the key features, in addition to being viscous, is that the melting point is much lower of different species, short species, long 3,000 kilo base pair duplex, that the melting point is reduced. So that's going to be important as we go forward. They're viscous, but they also reduce the melting point. And so in a paper published in Nature Chemistry in 2017, we were able to show that we could coat a strand with 30 tumors, 11 30 tumors, and we could ligate those on the template in glycholine. So if we had just aqueous buffer, we would not be able to make a copy. But we could coat and transfer the information, clearly showing templating by leaving out different of these oligomers, and perform that information transfer on a long template in the presence of the complementary sequence. So I thought it'd be nice to show Christine He, the graduate student of He in Nature Chemistry 2017, was the Sudduth Award winner in 2017. So some of you may remember that. And Christine also is a talented artist, and so she used e-painting to generate this cover and of Nature Chemistry. And he also freelanced for us to create the Georgia Tech Astrobiology logo. So her work lives on in Georgia Tech Astrobiology. So we thought this was a really promising approach. And, but we had one problem is that this glycoline is miscible with water and unreactive. So those are both good features. However, not really prebiotic. So the glycerol is, but the choline is really not so much. And also, we had a challenge in implementing multiple rounds because once we added divalent cations to actually stitch together the DNA RNA, it was no longer soluble. So there were some challenges. So going forward, Adriana Zoya Colinas, who recently completed her PhD in the center, has been taking forward a more prebiotic solvent, urea acetamide water. And this is a solvent that we've been using in the center for a number of years. And so she has been able to show that we could copy both complementary strands of hundreds of base pairs, and to do it over a wide design space here. The blue represents with a replication, both with and without salt, over a wide range here. So it shows that this is a very robust solvent system. And Adriana has also been able to show that we can ratchet this forward over multiple cycles, overcoming the problems that we had with glycoline and divalent cations. So here we can see over 5 cycles how we can produce almost linearly, really ratcheting forward these species. The 320 is the longest species, but we can see all of them are continuing to progress forward here in terms of both strands. So really being able to replicate both strands of a sequence, a long structured sequence, those are all really major accomplishments and advances to this overcoming of strand inhibition. So as I mentioned before, working in the center has really been fun. We've taken some particular directions, then they've taken us in surprising new directions, and then sometimes back again. There was an earlier idea, Irina Mamajanov, who I already mentioned, had been looking at non-aqueous solvents to drive condensation polymerization to help solve the water problem. Whereas for strand inhibition, we were considering using viscogens like polysaccharides and adding them to aqueous buffer to slow reannealing. But then we were kind of cooking the sugar, and so we had to go to too high of a temperature. So then we started using the small molecule non-aqueous solvents instead. And then we thought that really the key point would be that we could slow down the long strands diffusing and get the short strands to kind of come in and make the copies. But we had a much bigger effect than we expected. And so then we figured it was probably due to folding, and that these viscous environments could help with that. And so while viscosity may be an important factor, what we've been considering now is that the lowering of the melting point may actually be, maybe even more critical. And that's something that's consistent across the viscous glycoline, but the non-viscous urea acetamide water solvent. So with that, I'd like to conclude my presentation. I think throughout this research in the center, working with this amazing team, we've been able to get some insights into how peptides and Depsipeptides may have been really present, ubiquitous on the early Earth, challenging the dominant RNA world hypothesis alone. And we've been able to show how these condensation polymers may have been selected in life because they could evolve in water, but also how non-aqueous solvents can help drive selection for folding and function. And finally, although maybe it's frustrating, sometimes it can be necessary to take one step back in order to take 2 steps forward. So with that, I would like to again thank all of the members of the Center for Chemical Evolution, and to really acknowledge the work of the 3 PhD students whose work I really talked a lot about today: Sheng Yu, Christine He, and Adriana Zoya Colinas. Many wonderful collaborators in the center, including Nick Hudd, Ram Krishnamurthy, Joe Shork, Facundo Fernandez, Charlie Leota, and many thanks to Christine Conwell for keeping us all moving forward together, even though sometimes it was a step back and a couple steps forward. And I also really want to just acknowledge and celebrate all the work of the Education Outreach and Diversity team within the center. Our two directors, Mesa Salida and Chris Parsons, for the last 10 years. This wonderful mural painted by Christine He and David Fiallo, another graduate student in the center, in partnership with the Latin American Association in Sequoia Middle School. Antonio Lascano is right here, you can see. And I'd be happy to take any of your questions. Thank you.